So it's my pleasure to introduce Adil Razi, who is currently an associate professor at the Turner Institute for Brain and Mental Health at Monash University in Australia, and he heads up the Computational Neuroscience Lab. And he's been doing a lot of work sort of bridging engineering, physics, machine learning, and brain connectivity, brain structure, brain function. And he's done a lot of work with dynamic causal modeling, and he'll be talking about some of that today. And it's really great to have him here, especially given that it's 4 a.m. where he is, and he's also started his vacation. So he's very dedicated, and we're looking forward to hearing what you have to say. So without further ado, I will let you take it from here, Adil.
Thanks a lot, Vince. Thanks for the invitation. It's great to be presenting to you. So I will be talking about dynamic causal modeling of our brain function. So as Vince just introduced, I'm heading a computational systems neuroscience laboratory at the Turner Institute for Brain and Mental Health, and we are based at Monash University in Melbourne, Victoria. Okay, so let's start. Just a bit about what do we do. So we basically combine neuroscience with artificial intelligence and machine learning. So we basically develop generative models, and we are basically interested in brain dynamics. So what we actually do, so we investigate how different parts of the brain work together to process information in biological systems such as brain, and we are interested in how the brain is internally connected. And how do we do that? We do that by using a multidisciplinary approach where we measure the brain responses using multimodal neuroimaging, which includes functional imaging as well as structural imaging and EEG and MEG as well. We use machine learning algorithms, computer science mostly. Then we also use statistics, which is where the main thrust is in using Bayesian inference, and that's combined with dynamical systems, which is basically physics. So this is what we do. We combine all these measurement technology with theoretical and mathematical constructs from several areas to understand what the brain, how the brain works. Okay, so let's start. So so when we start with this, so I should start with how do we what are the three different kinds of brain connectivity. So the first one is structural connectivity. So as the name suggests, this is the physical presence of axonal connections. Usually it is, as I am showing you here with 3 brain regions, it is undirected connectivity. Although we have recently done some work with Andrew Zaleski's group where he has used some advanced analytics approaches where we can infer the directionality in a structural connectome. However, that's not the focus today. Then the second one is functional connectivity that most of you know very well. So functional connectivity is about statistical dependencies between bold time series. So as you could see again, there are 3 brain regions and they are connected with each other. However, this connectivity is defined by correlations or some other variants. We will talk about this in a lot more detail a bit later. But again, there is no directionality, how the information flows between these neural elements. The third one, that's the main focus of my talk today, is about effective connectivity. And effective connectivity is defined as the direct causal influences between neural populations. So this is important because this is not at the level of bold signal, but rather we have a generative model and we infer the connectivity between neuronal populations. Yeah, so that's important, and that will maybe make clear later on when we talk about certain applications, especially in neurodegenerative diseases. So that's the three kind of different kind of connectivity. So let me now go into something which we talk about the relationship between functional and effective connectivity. For this, I would like to show this as a connectivity reconstruction problem. So consider this as simulations. So these are all simulations, four set of different simulations. We have a generative model here at top, which has three elements, 1, 2, 3, and they are connected with each other with certain weights. Yeah, these give rise to three time series, and from these three time series, we can calculate the correlations. So you could see that with this structure, which remains the same across all four different simulations, we have the same structural connections, but the weights of these connectivity, this varies. And you could see that once we do that, for example, here, we change the weights a bit here. What you see is that this connectivity changes from 0.79 to 0.18, while the others are also changing. But for example, this one remains the same, 0.77 and 0.77. Yeah. Then we change the weights again a bit more, and this 0.77, which was very high positive correlation, converts into a very high negative correlation. And then we can also change these weights in a way that the node 3 is mediating the responses between the three nodes, and there is actually no correlation. So you could see that there are very different observations that we have, or the consequences that we have, but the causes, as you could see here, which are modeled by a generative model, they have very same structural uh, 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 pattern. So what we say is that this mapping between the effective connectivity, the top panel, and the functional connectivity, the bottom panel, this is many to one. So what does it mean is that this is a degenerate mapping, or what we call many to one mapping, or mathematically we call that this is an ill-posed problem, where there could be for a very different, so we can get many different patterns from the same structure. This is something that we have in many different problems, in many different areas, from brain engineering or neural engineering or brain connectivity to other areas in seismology and so on and so forth. So this is what we call an ill-posed problem. Now, to regularize this problem, what I would like to show you is what we call Bayesian solution or dynamic causal modeling. Here we show effective connectivity as a generative model. So consider this as sorry, this generative model. So here, what I'm trying to, so this is some graphics that I will show um, for the next few slides. So it's good to know what does it mean. And this probably goes to the heart of the main topic of my talk, which is about generative models. The generative models here, what we are saying is this, that let's say that there is some endogenous fluctuation or an external stimuli. It could be some perturbation which perturbs the dynamical system which we use as a to model the brain. Yeah. So here we have this first order differential equation, x dot equal to function of current state x. some input U. It could be endogenous fluctuation, as in the case in the resting state fMRI. So these are neural fluctuations, or they could be external stimuli as well, as is the case in the task fMRI. And then the model, then these theta, which are the model parameters, which are the connectivity between different neural elements. So this is a first order differential equation which we are using to model the brain. Yeah, and then what happens? This input comes in. It perturbs this system of equations, and then what we get is it goes to the hemodynamics, and then what we get is y, which is nothing but hemodynamics plus neural activity and plus some measurement error. And this y is our bold signal. And from this y, we can calculate functional connectivity, which becomes, let's say, our data. Yeah. So what we have this generative model as where we have this effective connectivity theta here, which I show as a sparse matrix, and this is what we call as a causes. So this is an important point here. Sorry. So we consider effective connectivity as a causes and functional connectivity or the observations as consequences. So what we are having here is that the effective connectivity causes consequences or the functional connectivity, which we show here using a generative model. However, remember that this mapping is not one-to-one, is many-to-one. It's an ill-posed problem, as I was talking before. Okay, so this is a generative model, but however, in practice, what we have is the other way around. What we have is that we have bold signal or the functional connectivity, and from this we want to infer what was actually happening inside the brain. And this is what we are trying to do using functional connectivity or some other data features which are second order statistics which represent functional connectivity variance or functional connectivity itself here, like a covariance function here. And we use this as a data feature, and then we try to infer the effective connectivity of these parameters theta. And this process of going from functional connectivity or from consequences back to causes is what we call a model inversion, or in this case, because everything will be done in a Bayesian framework, this is what we call Bayesian model inversion. Yeah. So what we are doing is this: that when we are doing this model inversion, we have a cost function which we define as a model evidence, which we minimize, or this is what we call a free energy. So free energy is a function of our data sigma and our and some other precision and other model parameters, and this is what we are trying to minimize when we do this optimize. So what we're doing is this: that we are converting this in an ill-posed problem as an optimization problem. So when we optimize this problem, going from our consequences from using functional connectivity, and we try to infer our effective connectivity, that is theta, what we get once we optimize this cost function, we get two quantities. One is this: that we get a posterior density. So posterior density are the model estimates or the parameter estimates P theta given data sigma for a certain model. So this is our posterior. So this is what we are interested in. So important point here is, again, here is this: that because this is all Bayesian, our parameter estimates are not point estimates; they are distributions, and these are shown by this expression. And when we do this optimization, we not only get our posteriors, we also get what we call a model evidence. And this model evidence, which we hear usually it requires integrating very high dimensional integrals. So usually we cannot calculate it exactly, rather we calculate a lower bound to this, and this is what we call free energy. So so what we get is a free energy. So free energy is in a sense, you can say it is a number attached to that model, which itself is nothing, but it can be very useful when we do a model comparison. So it's basically provide a ranking or a score to a model. So remember that remember that when we do this model inversion, going from consequences to causes, there could be one cause, but it could become from many different combination of the causes, and this is what we call an ill-posed problem. And here, what we are doing is this: that we are using model evidence to solve this ill-posed problem. So, for example, here I have done the model inversion. I get many different possibilities that I can choose from, many different combinations of my connectivity parameters that would give me the same consequence. But what I have here is this: that every model has a log model evidence or the free energy with it. So here, what I'm plotting here is for every model, I'm plotting their model probability, and also what I'm doing here is what I'm showing here is the probability of a certain model given data, and using Bayesian model in comparison, we see that this model, let's say this model number 21 or 22, it has the highest probability. So we say, okay, this model which has the highest model evidence is the best model or the preferred model compared to the models that we have tested. Yeah. So what we say here is that this is the we don't say that this is a true model or a ground truth, but rather what we say is that this model is the best model among the models that we have compared. Yeah. Usually what happens is this: that this was the case where one model had a very high model evidence. Usually this is not the case. A lot of models which are similar, they have a very similar model evidence, and in that case, what we usually do is this: that we use a Calm's window and calculate a Bayesian model averaging within that window. And this is what we are showing here. So Bayesian model comparison, yeah, and then we use a family of models to calculate an average model. So this is briefly the whole procedure of model inversion and model comparison, which rests upon free energy minimization and model evidence, which come which is the free energy, which is the lower bound of model evidence. So this is the whole framework of solving the connectivity reconstruction problem that I initially showed, and this framework is what we call dynamic causal modeling. Okay, so here one very important technique or technology that we have recently developed is what we call a Bayesian model reduction. Remember that if we go back to the previous slide, there could be, while I'm showing here only a few models, there could be millions of models that we need to actually compare. And every model needs to be, for every model, we need to do a model inversion. So this technology that we call Bayesian model reduction is a more recent work, which allows us to score or invert models, millions of them, very, very quickly using something what we call a Savage decay ratio. So for this, if we know the posterior and the free of a fully connected model. So let's say we have a model where we have what we call a parent model, and we invert that once, and then what we can do is this: that we can score or find the model evidence for every nested model or the reduced model within that parent or the fully connected model. Using this technology, it takes few minutes to score millions of models, and then we can find a family of models using Bayesian model averaging. This is something that we use now very extensively in our framework to model a large number of to score a large number of models. Okay, so this is again one more point that I wanted to make is about the model evidence and the account principle. The free energy that I was talking about, which is actually nothing but the lower bound on the log model evidence, we can decompose this free energy into two components. One is the accuracy, and importantly, the second component is complexity. So unlike other methods of model inversion or model fitting, here what we are doing when we are minimizing free energy, we are finding a balance between accuracy and complexity. So the main idea is this, that we wanted to find a model which is simplest but the most accurate explanation of the data at hand. So that's basically what we try to achieve with our method. And what I wanted to show here is this, that this free energy is a function of data. So what sort of data that you use? For example, I have functional MRI data. Functional MRI data is usually modeled, as I was showing before, using a first order differential equation. Here I have shown this as a Taylor approximation, which is a bilinear approximation here. Very simple approximation, not many model parameters. fMRI models are very simple. And what we see here is this, that on the x-axis we have the complexity, and on the y-axis we have the free energy or the model evidence. So we see that at certain point we have this nice spot here. But if we look at EEG data, the EEG models, and I will show later on, these EEG models are very complex. They have large number of higher order differential equations, many models, thousands of them in some situations. So very complex data. So you see that then they have this model evidence at another point, the sweet point is here. So what I'm trying to show here is this, that the evidence is afforded by data. It is a function of our data. Yeah. So the main point from this slide is this, that with the free energy, what we are trying to do is this, that find out the simplest model, which is an accurate description of the data. Yeah, and this is what we try to get by minimizing free energy. Okay, so let's okay, so yeah, so this is a bit involved slide, and I don't want to scare you away with this slide, but it's very simple in the sense that what I'm trying to show here is this, that if we want to calculate connectivity, we have to have, or the effective connectivity, we have to have state space models. Now, those who have background in signal processing, they know, that things. This is an instance of a state space model or a dynamic causal model. Dynamic causal model is a state model. So what we are showing here is this, that if we start with a state space model or a dynamic causal model, we can actually drive. We can start here at top, and then with certain approximations. For example, we can calculate vector autoregressive models. We can calculate convolution kernels, and from these we can calculate cross covariance. We can calculate cross correlation, and so on and so forth. So all of these can be exactly derived or calculated if we start with a state space model. These are time domain representations, these two columns at the extreme sides, and then the internal two columns, they are equivalent but spectral representation. So we can take a Fourier transform, for example, of a convolution kernel, what we get a convolutional theorem, or we calculate a Fourier transform with cross covariance function, and what we get is a cross spectral density, and from which we can calculate coherence, and so on and so forth. And also this, that you can see that these last two rows, they are second order statistics, while the upper two rows, they show first order statistics. Important point here that I want to make is this, that for this to happen, you have to start at the top, and this is a one-way street. If you start at the top with a state space model, you can calculate, for example, these are all different variants of functional connectivity, right? Coherence, cross correlation, causality, so on and so forth. These are calculated from the state space model, but this thing is one-way street. You can only go from top to bottom. But if you have a cross correlation function, and if you didn't have the state space model, you cannot calculate what's the causes, yeah, or the effective connectivity. So that's basically this slide is talking about. Okay, so let's move on to some more recent developments, and I want to also show a couple of applications of the modeling as well. So let's start with what we call large-scale spectral DCMs. So what I'm showing here is this, that we wanted to look at whole brain connectomes, which are, for example, in this case, I have 36 nodes network, which I have taken from Marcos Reikel's paper, which are 7 different canonical networks. Each of the network has certain amount of certain number of nodes, and total number of them is 36. And what we are seeing here is a what we call a causal connectome. Here you can see the directionality, and you can also see that there are 2 different sort of connections. One is the green connection, and one are red, and they are the greens are the excitatory connections, and the red ones are the inhibitory connections. So this is some information which is more than what you can get from a functional connectivity connectome, because there you don't have a directionality and you don't have the heterogeneity of the connection type as well. So what we did is, so in order to invert these nodes, these connectomes, there are two competing methods. One is what we call a stochastic DCM, and the other one is what we call a spectral DCM. Stochastic DCM is basically solving everything, the system equation or differential equation that I was showing you, and doing the model inversion in time domain. And that's something what we call the stochastic DCM. It is a very sophisticated scheme based on generalized filtering. It is also called triple estimation problem, because here we have 3 different sort of parameters. Here the states are also changing over time. By the way, this is for resting state, which means that the states have neural noise in it, so which are perturbed by the neural fluctuation, which means if we try to solve the system of equations in time domain, we have to estimate the states, we have to estimate the model parameters, and we also estimate the model's hyperparameters. So there are 3 different quantities that we're estimating, and that's why this is also called a triple estimation problem. It takes a long while. So for this, in order to invert this 36 nodes network, I would require on a large cluster, it would take about a month for a single subject. It's very, very time consuming estimation. In comparison, there's a spectral DCM, which solves everything in frequency domain. We use certain tricks to expedite the inversion, and we can actually invert such a model within a day or within 3 hours. Okay, so this is the two schemes. Here we are showing that there are prior eigenmodes and the free energy, and what we're trying to show here is this, that about using, when we use about 10 modes, we can get similar free energy. Just a sort of a validation that we are providing here. So what you get is these connectomes with the spectral DCM, where you have these green connections, as I said before, excitatory connections, inhibitory connections. Different node colors are showing different brain networks, so on and so forth. So this is what we call a causal connectome. I wanted to show you some examples how we apply this. So one of the very early examples was I wanted to show was about aging and lifespan, using the lifespan data set of Cambridge Connectome, Cambridge Center for Advanced for Aging Network, CAMCAN. So the problem that we are looking at in this one is this, that this is very well known. Brain has different functional networks, and for example, two of them I'm showing here: default mode network and causal attention network. And as you could see, these are anti-correlated. Yeah. So this is coming from very seminal work from Power et al. Then there was some work done by Giolix et al., where they showed that the young connectomes and old connectomes, where they look very different. As you could see, that young connectomes, they are very tightly coupled within a network, and there's a sparse connectivity between networks. While as people get older, as they age, they have less within network connectivity, and they have more between network connectivity. So this is what we wanted to look at using the spectral DCM. Important point that I want to make here is this, that especially in terms of for neurodegenerative diseases or aging studies, the functional MRI or the bold signal itself is a very complex signal, which consists of some neural part, and then there's a hemodynamics, and they are coupled together using a neurovascular coupling. I will later on show you a much more involved um, uh, way of modeling the bold signal later on. But this is a schematic which shows that fMRI bold signal. For example, if it is task, or if we don't have the task of a resting state, composed of these three components plus some observation noise. Important point here is this, that with the it will differentially effect on neural and hemodynamic and neurovascular coupling. It's not like the age it has the same effect on all these components, but it's a differential effect. This is a very nice description from Tismanov et al. By recent work. So the idea behind showing this is this, that if we are using a functional connectivity measure using bold signal, and you find some differences in functional connectivity, especially in neurodegenerative or aging populations, you don't know that if the differences that you are seeing, they are because there are differences in hemodynamics or neurovascular coupling, or what we call the brain plumbing is getting old, or it is actually some neural changes of interest. Yeah. With DCM, what we are doing is this, that because we have a generative model, we can actually um, we can actually uh, compartmentalize these three different contribution and also the noise as well. And this is what we did in this example. We used three canonical networks: causal attention network, default mode network, and salience network. There's quite a lot of evidence that DMN and DAN interactive, these two network connectivity this is disrupted in older adults. And DMN and DNN and DAN interactivity is mediated by the salience network. Yeah, and salience network itself, its functionality declines with age. And we wanted to see what happens with these two networks when we try to tease apart different contribution to the board signaling using DCM. So what we did, for example, here we are showing between network connectivity only. So there are some other results for within network connectivity as well, but just for the sake of in the interest of time, I'm only showing you the between network connectivity results. So as you could see here, we have effective connectivity parameters here. We have neuronal parameters. This is resting state. So we had random fluctuations or what we call neural fluctuations. They are also modeled here. They're modeled by two parameters using basically they are modeled as a power law distribution having two parameters, amplitude and exponent. Then we have a hemodynamics, and they are parameterized by few of the different sort of quantities like quantum decay and epsilon. So here you could see that then you can compare these as between people who are young and old or middle-aged, and you could look at the effective connectivity. Yeah. Here we have used chronological age and predictive age, high correlation between them. Importantly, what we did is we use canonical correlation analysis to associate the connectivity with cognitive profile. So there were a lot of different behavioral scores that we had, and we had connectivity between network, and we did use canonical correlation analysis with a multivariate analysis. This is what we call a helio plots. So here, these bars, they show the individual connections contribution. If they are going out from the circle, they are positive. If they are going in, they are negative, so on and so forth. So we see that this association is pretty significant. When we also did the regression analysis, we tried to plot. What we did was to, there were about 600 subjects, by the way, in this data set. It is a lifespan data set from I think the age of 10 or 12 to about 85 years of age. And these three, the whole lifespan data set was decomposed into three different age groups, young, middle, and old. And as you could see that people, who, for example, who were old, they had more between network connectivity to get the same performance level. And this was very significant, as you could see for the green one. So this is a nice example, and I didn't put those results here. When we use functional connectivity for this prediction, the results were not very significant, or the discrimination between these three age ranges was also not very clear. And the reason because is this, that with the aging, with BOLD, with FC, putting together all those different contributions, while here we are only looking at with DCM, we are looking at connectivity which is between neural populations. Another example that I wanted to show was we wanted to look at these three networks is related work done by Yuan Zhu, Chinese Academy of Sciences. We used these again three large scale networks, core default mode network, signal network, and DAN. This is the usual functional connectivity pattern that you see, that DMN is anti-correlated with salience and DAN. Yeah, so you see this well-known functional connectivity pattern. What we wanted to know is how it looks like in terms of the causes or the effective connectivity. So here what we have is this, that now you see that the symmetry is broken. And what you could see is this, that first of all, the regions which belong to the same network, they have grouped together, and they have within group they have uh, uh, high connectivity. This is a different data set. This is not a lifespan data set. This is the data set of twins which were all young. So it's a young connector. What we see is this, that the task positive or the salience and the dorsal attention network, they inhibit the DMN, while the DMN excites the salience network in DMN. And salience network sits at the top, at the apex of the hierarchy. So this is what we are showing here at the top. These are excitatory connections at the bottom. So at the top are the inhibitory connections in cyan, and in gold color, these are the excitatory connections. Here we look at at the network level connectivity. So this is a sort of an application that I wanted to show. I want to quickly go through another example in the next 5 minutes, a new recent development, which is what we call a multimodal DCM, or what we also call Bayesian model fusion. The important point about this application is this, that this is about fusing different modalities. For now, I have been talking about functional MRI only. Here, what we want to do is to connect or fuse electrophysiological signals like EEG with functional MRI. An important point here is this, that for this, you don't have to do the concurrent acquisition, but rather these two modalities can be separately acquired in the same subject or separate subject, but we can then use our modeling approach or the generative model to fuse them together. Yeah. But for that to happen, what we have to have is the same generative model for both of them, or the same neuronal model, which I'm showing here by X, as I previously showed you, as the first order differential equation. Let's say this function f. So what we will be doing is this, that we have created or developed a generative model based on canonical microcircuit, which will be the same for both modalities. Yeah. So once we have a same a neuronal model for both modalities, both for functional MRI and also for EEG, then we can just change the forward model. It could be a lead field for EEG and hemodynamics for fMRI, and then we can fuse them. We can do leaf updating and so on and so forth. I will show you quickly what we do. But importantly, what I want to introduce is the generative model for this scheme, which we call a canonical microcircuit. Again, pretty there are a lot of parameters and a lot of equations here, but it's very simple. Again, what we have here is two sets of differential equations. One, this is neural dynamics, and the second set of differential equations which shows hemodynamics. And these two sets of differential equations are coupled by using are coupled by a linear neurovascular signal. What happens is this, that experimental input perturbs the neural dynamics, and these neural dynamics are modeled by a canonical microcircuit. This canonical microcircuit composed of four neuronal populations. Number one, which is spinal skeletal cells. Number two, superficial pyramidal cells, inhibitory neurons, and deep pyramidal cells. Yeah. Each of these populations has two states, P and Q. So each population is modeled with two hidden states, and these are modeled by a second order differential equations. These equations model the depolarization of each population, which I'm showing here with these, where you have an afference, you have an experimental input, and you also have an afference from other populations, or from population in the different region. What's happening here is this that we also have, as you could see, these different populations are intrinsically connected. These pink color connections, the inhibitory connections, blue colors are color ones are the excitatory connections, and the green colors are color projections are the collateral projections, astrocytes, and then the neurovascular signal, and this induces the hemodynamics. So this is a canonical microcircuit, and once you have that model both for fMRI and EEG, you can have something like this. So this is a scheme. I'm mindful of time. I will just try to finish in a minute. What we have, let's say you have acquired data separately, one for EEG and for the fMRI data as well. Yeah. For example, it could be a HCP data set. It could be MET data as well. So it could be HCP data set where you have both fMRI and EEG and MET data. So what you do is this, that you use canonical microcircuit, use MET data, do the model inversion, and get your posteriors. Then you take those posteriors and you put them as a prior in your canonical microcircuit for fMRI. Remember, this model, this neuronal model or generative model here is the same for both of them. So use the posterior from EEG, use as prior for fMRI, and do your inversion, and then what you get is a model parameters for fMRI, which will include all those model parameters that I was showing you in the canonical microcircuit, plus you get hemodynamics as well. Yeah. So this is and that's how you can fuse two different modalities without the problem of acquiring data concurrently. Yeah. And this one quickly, this is where I'm talking about dynamic effective connectivity. Again, a bit involved, but again, very quickly, I can describe this. This is the last slide. So, for example, again, this is done on HCP data set. So there are 4 sessions. For every session, you can do windowing. Till now, all of the work that I showed you assumed that for the whole session, the effective connectivity parameters, they are time invariant. They don't change over the length of the experiment. What we are doing is this, that we are dropping that assumption. We do a window. We for every window, we invert and our model, and we get our model parameters. We move this window. So as you could see, this is over window. So for example, here we have 12 windows or 11 windows for every subject. So this for every session, sorry. And each block is a connectivity parameter matrix. Then when we combine them using a line matrix here, we use a discrete cosine transform, get this into a session level EC, and then we do another. I'm not going into details of parametric empirical bills here. We don't have time for that. But in the end, what you get is an individual. You get a group level dynamics modeled. So this is something, again, not very old. We recently did the extension. With this, I would like to thank all my collaborators, especially Carl Fiston and a lot of other people at the UCL methods group, and also quite a few people, close collaborators at Turner Institute. With that, I would like to thank everyone. I'm happy to take any questions.
Great. Yeah, thank you very much. So just as a reminder, if folks have questions, you can enter them in the chat, or you can say, please unmute me. I'd like to ask the question myself, and we'll work with you either way. There is one question so far, which is, would you please comment on the replicability and reliability of dynamic causal modeling across individuals? For example, do we see stronger Dan to DMN versus DMN to Dan across all subjects using the DCM model? That part first, yeah.
Yeah, that's a very good question. So there is there is a variability across subjects. What we do is this, that we have, and I didn't have the time to introduce this. We have a method, what we call parametric empirical base. This is, by short, we call it PAP. This is something that we use at the group level. But it is very useful in the sense that, because what happens when you do inversion for a single subject and then you do these independent inversions for every subject, what happens, it could be that every subject, or some of the subjects at least, they have a local minima and they're stuck there. Yeah, and then this would result into quite a bit of variability. But using parametric empirical base, what we could do is this, that we do the first round of inversions for every subject independently of each other. Then what we do is this, we take the group level posterior, and then we use this as a prior for every subject. And then those subjects which were stuck in a local minima, we can use that group level posterior to rescue them. And this increases the reliability of the subject level estimates. There are quite a few papers which look at the reliability and also replicability of DCM. Some of the papers we recently published. One of my PhD student, Hannes Almgren, he had a couple of papers specifically providing validations for DCM. Later on, I can provide some of the links to those papers. In task DCM, Stephen Frazel has done quite a bit of validation work as well.
Okay, great, thanks. Dipanjan Ray asks, is there any DCM for multi-brain effective connectivity?
Yes, there is. There is. I would be able to forward you the recent preprint, although I know little about it. But there is now multi-brain DCM as well. This is a work from Elda Bilic. I'm not sure if I'm pronouncing her name correctly. She's a new PhD student working with Carl at UCL in London. I was not part of the project, but I can forward you or get you in touch with her if you are interested.
Great.
But there is a multi-brain DCM, very recent. I think the preprint was done last month.
Okay, sounds good. Another question from Dan Handworker says, as you note, the causal estimates for fMRI using DCM rely on a model of neurovascular coupling, but based on observed data, we know that the amount of variation in neurovascular coupling varies more than modeled. How would mismodeling affect results?
Yes, so I mean, this point I spent quite a bit of time talking about, in terms of, especially in terms of aging populations, because this is something important when you try to apply DCM to neurodegenerative diseases where, the neurovascular coupling would be differentially affected. Yes, so I mean, this is a concern, but we, so there are, for example, we as I discussed before, in terms of modeling efficiency, we do try to use parametric empirical base to have better model fitting, not only using independent data sets, but rather group level data and do recursive modeling, and so on and so forth. But this is something which is a limitation, I could say. I should say, and requires a bit more, further work on this. And there are some of the modeling initiatives which are looking at it.
Okay, thanks. And Kauser Abbas asks, in the Bayesian model inversion, is FC treated as a single object, or are all edges treated separately?
Yeah, so basically, FC, if I talk about spectral DCM, so what we use is quasi-spectral density. So this is what we are fitting, and we calculate it. We calculate quasi-spectral density, and then we use this as a data feature. I'm not sure what do you mean by a single object or all ages are treated separately. I mean, so you will have, you know, as many edges, you will use the cosine function for them separately and use from that to calculate your spectral densities and fit them. So I'm not sure. I should say,
Right. Yeah.
if I understand they are different. Yeah.
I guess it can be clarified, but my guess would be maybe is it treating the entire network as a whole and comparing it cross networks versus? It looked to me like you were dividing it up into subnodes, right?
Yes. If I understand what it is, yeah. Yeah, so it's not
Kauser, feel free to. Yeah, sorry, I was just going to say to Kauser, feel free to ask if he didn't get your question figured out. We're trying to guess what your. Yes, let's unmute him. Armin, can we? That'll be easier. So.
Yeah, I understand what you mean. Probably we are not fitting for every age, for every edge. We do we consider it as a single subject in a sense. So we do one inversion and we fit all of them together.
Hi. Well, first of all, I have to say, really nice presentation. But I would like to clarify just one thing. I think your last comment is very relevant. What I meant to ask was, even when you're treating the whole functional connectome as a single object, are you vectorizing it before doing the spectral decomposition, where each edge is treated as a separate feature?
So what I will be doing is, first of all, I'm not using, in a sense, correlations. I'm using the whole cross covariance function, yeah.
Okay.
And then there will be a bunch of, so if I have a network, I will have a bunch of these covariance functions, and those I am fitting. So what I'm fitting are all of those quasi-spectral density that I'm fitting. So I'm not sure if this clarifies anything further.
I guess what I'm asking is that functional connectome being a covariance matrix, all the values in the matrix are inherently interrelated. So yes. That's the only assumption that I was actually referring to.
Yes, of course, yeah, definitely.
So if that assumption is being broken or not, that I think that's the end question, I guess.
No, of course, all the dependencies or independencies are. Yeah. So they are not considered as a separate thing, and we don't look. So it's a multivariate optimization, not univariate optimization. That's it.
Okay. Thank you.
Okay, great. Let's have maybe our last question. Oh wait, no, there's a couple more here. So one is, how many spatial modes? This is from John Griffiths. How many spatial modes do you find are generally needed to explain resting fMRI data? And do you interpret data in terms of the spatial patterns of individuals' modes? Allow them to vary across subjects. Yeah.
Yeah. Thanks, So if I have a smaller network, for example, let's say 6, 8, or 10 nodes, then I would use all the modes. Yeah, so I don't do any mode optimization. But if I have larger nodes, and there was one slide where I was showing, maybe I could go back to that slide. This one. So in this one, what I was doing, because it's a bit larger network, what I tried to do was to use only few modes. So this is where I get a lot of efficiency. For example, here, instead of using all the 36 or 35 spatial modes, I'm only using 6 or 7 of them, and that lets me efficiently invert my model. So it depends. And in this situation, what I'm showing is this, that if I use all the modes, or if I use 6 or like 8 or 10 of them, the model evidence remains, it is flattened. So there's no point in using a lot of other modes. I can just use few of them, and my inversion would be equally good in terms of model evidence. If that's answers your question.
Okay. I will, in the interest of time, let me see. I'll try to just summarize one more question. But before that, Heng Bahi asked if for multimodal DCM we need to synchronize fMRI and EEG. And I think the answer was no, because you're using this generative model and fusing at another level, so they can be separately collected.
Exactly. Exactly.
Yeah. And Kamal Premaratni, sorry if I'm butchering your name, we would like to replicate the data you use. For example, the data in the 2014 Network Neuroscience paper, you get an authorization from the administrator message. Is there a way you can share the data so your results can be replicated?
Of course. So which one? So is this one 2018 Network Neuroscience paper?
It's a 2014 paper.
2014 paper.
Yeah, I'm not sure, but maybe they can contact you directly.
I mean, everything that we have, there are a couple of papers in 2014. 2014 paper where we introduced this one, this spectral DCM, is a single subject, and I can send you the data set.
Great. Okay, well, thank you. So we should let you get to your day. You're beginning your day of having a great vacation. Enjoy the island. Thank you so much for joining us and letting us feel like we're part of the island and all connected here. Thank you again, and we'll wish you well. All the best.
Thanks a lot. Thanks everyone, and thanks Vince for inviting.
Just a reminder to folks that we'll follow up with the Slack channel, and the talk will be posted. And if there are questions, further questions, we can follow up in Slack. So, and we'll reach out to you.
I will be there to answer anything.
Okay, we'll help. Yeah. All right. Thanks so much.
Thank you.
Bye-bye.
Bye-bye.